All right, we've been talking a lot about genetics and how our DNA can lead to certain traits, but we haven't talked about how does this happen? How does DNA and the code in our DNA lead to a certain hair color, skin color, or eye color? What happens to get the code to be expressed? So in this picture here, we can see that there are many different traits displayed for hair color and skin color and eye color. And the interesting thing is, every one of the cells that these people have contains the genes or the genetic code for each one of these traits. However, only melanocytes or certain cells that have melanin in them make the pigments. So how come some cells make these certain pigments, or melanin, which is responsible for skin, hair, and eye color, and other cells do not? How are these codes turned on and off? How are these certain traits expressed only in certain cells and not others? We're going to talk about that in this chapter, and also how this code is expressed, how the code is turned into a certain protein, or in this case, melanin, which is a certain protein. And this is going to be chapter 12. The first thing we have to talk about is how do they discover DNA, or genetic material? What is this stuff? So the first thing that was discovered are these chemicals, adenine, guanine, thymine, and cytosine, and we represent them with the letters A, G, T, and C. And they found out that every single species had adenine, guanine, thymine, and cytosine. And Chargaff's rule says not only did every species have these chemicals, but turtles might have a certain amount of adenine, and every single turtle would have the same amount of adenine, the same amount of guanine, the same amount of cytosine, the same amount of cytosine, and that amount was different from every cell of a human. Human would have a different amount of adenine, guanine, thymine, cytosine, and it seemed to be the same throughout an entire species, and each species differed from the other. And not only that, but each species had an equal amount of adenine and thymine and an equal amount of guanine and cytosine. So if you look at this chart over here, Homo sapiens, humans, we had 31, what is this percentage? This is not, 31% adenine and 31.5% thymine. That's very, very, very close. Same for guanine, 19.1, 18.5, very, very close. About the same amounts of adenine and thymine, and then the numbers for guanine and cytosine would be about the same. But they differed between the species. So they knew this had to have something to do with why certain species were the way they were, had the certain traits they did. They didn't know the shape or what it exactly was yet. And then we have Rosalind Franklin. She studied the structure of DNA. She's trying to find out what does this genetic material look like. And she used something called X-ray diffraction photographs and got this picture right here. It just looks like an X, right? And she wasn't sure what to do with it, but she was the first person to actually photograph DNA. And what it is, DNA is a helix, so it's like a ladder that's been twisted. And you can see this is looking from the top down. It's like looking at the top of a twisted ladder. So you can see it kind of spirals in there. So she showed her picture to some colleagues of hers who showed the picture to someone named James Watson without her permission, and he actually understood what this picture meant. And he took her picture without any credit and went on to produce the first structure of DNA. And Watson and Crick, they actually did this together. They published their paper and actually received a Nobel Prize. And what they found out was the backbone or the rungs, not the rungs, the sides of the ladder. If we think about the twisted ladder that DNA is, think of the sides of a ladder. Those are sugar and phosphates connected together. You can see phosphate, sugar, phosphate, sugar. And they're actually opposite. So this one would be sugar, phosphate, sugar, phosphate. And then the rungs of the ladder are those chemicals we were talking about, the bases, adenine, thymine, which go together, and guanine and cytosine, which go together. And that explains why Every time there was a guanine, there was a cytosine, so they'd be the same percentage. And then adenine and thymine go together, so they're the same percentage. And another thing that's interesting, we used to, back in our bonding chapter, the chemical bonding, we talked about covalent bonds and hydrogen bonds. So the sides of the ladder, the sugar phosphates, those are going to be covalent bonds, so they're pretty strong. They're not going to break very easily. However, the bases here, the adenine, thymine, and guanine, cytosine, the rungs of the ladder, they're connected with hydrogen bonds, and those are going to be a lot easier to break. So that's going to be important to remember later. And then this whole ladder is twisted into a double helix, is what we call it. Oh, and the fact that guanine goes with cytosine and adenine goes with thymine is called complementary base pairing. Okay, so they received a Nobel Prize in 1962 for discovering the structure of DNA, and they didn't give any credit at all to Rosalind Franklin. So that's and they're actually not the best characters if you research them, but they did come up with the current structure we still use today. Okay, so we've been talking about DNA, whole new cells being replicated, or the division of cells and mitosis. And we talked about interphase and mitotic phase of a cell. So mitosis is actually the division of the nucleus, nuclear division. But in order to have a whole new cell or divide the nucleus, we first need to copy the DNA. And if you remember, we kind of mentioned that DNA replication or copying of the DNA happens during interphase, so right before mitosis. We need to make more DNA because every single cell is going to contain the DNA. So let's go through how this happens. It's called DNA replication, and it's the process of copying a DNA molecule. So there are 5 steps that we're going to talk about for replication. First thing, the 2 strands of DNA, because remember it's double-stranded like a ladder, so we're going to take that ladder apart. It's going to unwind and unzip. Now the part that's going to unwind and unzip, remember those hydrogen bonds that were between the bases? That's where the ladder comes undone. It's like a snap-together ladder where the rungs of the ladder that you would step on actually come apart in the middle. That's the hydrogen bonding. So it's going to unzip there. And then we're going to have a protein that binds to the where it's been unzipped and holds it open. It's like a bookmark. And then we're going to put down new base pairs, and that's going to be not too bad because every time there's an A, we know a G goes there, and every time there's a G, we know an A goes. Oops, sorry, I did that backwards. Every time there's an A, we know a T goes there. And every time there's a T, we know an A goes there. And every time there's a C, we know a G goes there. One way to remember this is G and C look very similar, and they actually bond together. And here it is right here in case I confused you. A and T go together, and G and C go together. I just remember G and C look very, very similar, so they pair together. Okay, so we built a new strand. That's going to be a daughter strand. The whole new one is a daughter strand. And then they're going to proofread for mistakes, make sure everything's in there correctly. And then we're going to connect little pieces of DNA fragments together. So each one of these steps, each one of these 5 steps of replication requires an enzyme. We've talked about enzymes before. Enzymes do all the work. And enzymes usually always end in this ASE. So each one of these, DNA helicase, DNA primase, DNA polymerase, and DNA ligase all end in the ASE. Every time you see that, you know that it is an enzyme that we're talking about. You do need to know all these enzymes and what they do. They're very, very important. So DNA helicase is going to be the one that unzips. DNA primase is that bookmark that's going to hold it open. Make sure you put DNA in front of it too, because there's also RNA we're going to talk about later. Okay, DNA polymerase is going to be the builder of new strand halves, and I'll explain that in a minute. And then DNA polymerase also proofreads for mistakes, so it builds it and proofreads. And then DNA ligase is going to glue pieces together. The reason we need DNA ligase to glue new pieces together is because, well, let me show you on this next strand here. Here we go. Because in eukaryotic replication, the replication or there's something called a replication bubble. And that's where this entire process is occurring right here. So a new strand is being created at each one of these replication bubbles. And it doesn't just happen in one spot, it happens in multiple spots. So once they meet each other, we're going to need DNA ligase to glue the two pieces together. And there's actually something called an Okazaki fragment that I love to explain in class, but I think we're going to skip that now just because we have missed some time we're trying to make up. But if you do see Okazaki fragments and connect, those are pieces that also need to be glued together. Okay, so the glue is DNA ligase. It's going to glue these bubbles together. See, once they meet, glue them together. Now, another thing to notice here is that the green is this new daughter strand forming, and the blue is the original parental strand of the cell. And you can see when we're done, these two new strands of DNA, one half of this is going to be a parental strand, and one half is a daughter strand for each segment, each piece of DNA. So every single cell will have the original, or both cells will actually have the original parental strand and a new piece, a new daughter strand. That's called the semi-conservative model of DNA replication. I also want to touch on prokaryotic replication. It's much simpler, which is why it's used a lot in biotechnology, which we'll talk about in a little bit. So remember, prokaryotes, they do not have a nucleus, and they also have circular DNA. It's much simpler. So this circular DNA, replication only happens in one spot and continues throughout the entire thing on both sides, and ends up kind of unzipping to form two new circles, circular strands of DNA. Well, they're circles. And again, just like in eukaryotic replication, you have one parental strand and one daughter strand. Okay, so this is what we already talked about, where we have the different enzymes, but you can see them in action here. This is going to be DNA helicase unzipping. This is DNA polymerase, which is building the new daughter strand, DNA polymerase. Remember, there's two parts now. So we need DNA polymerase to do it on both strands. And then we have DNA, oh, we forgot, we skipped DNA primase. That's going to be the bookmark that holds it open. And then we have DNA ligase that's going to glue pieces together. Okay, so replication of DNA is really, really accurate. It's very accurate, but it still makes a mistake about once per 100,000 base pairs. It's capable of identifying and correcting the errors by proofreading, so it can say, oh, there's a mistake right there. G does not go with T. And so it'll cut it out and fix it. Put A with T. But sometimes it doesn't catch the mistake. Sometimes that G goes with T and it doesn't get fixed at all. Instead of fixing it, it will actually go and be a mutant. Oh, and the reason it's a mutant is because when it tried to fix it, it actually fixed the wrong one. So instead of changing there. Okay, let's look at what happened here. We have the original DNA strand. It's been unzipped and new daughter strand has been put in. And instead of pairing the C here with the G, the T has gone with the G. It's formed a little mismatched bubble right here that DNA polymerase is able to proofread and see, hey, that's wrong. It cuts it out, but instead of replacing this part right here, down here it actually replaced the G. So now instead of being GC, like it should be, it's AT. So that's a mutation right there. Most of the time, mutations are not advantageous at all. Sometimes they have no effect. Sometimes they have a very profound but negative effect. But very rarely they can actually be beneficial. And we're going to talk about what happens then in another chapter. All right, I'm going to stop this video here, and then I will start with the protein synthesis for you guys.